Dear learners, welcome to today's learning session, Deciphering Genetic Code. We know that there are thousands of proteins involved in the cellular activity of all living beings, and they are all formed from only 20 amino acids according to the information coded on the blueprint, what we call it as mRNA. The mechanism by which this four-letter DNA, that is the nitrogenous basis, is transformed into a 20-letter amino acid text is known as the genetic code. Deciphering genetic code is considered as one of the pivotal achievements in molecular biology, unraveling how DNA sequences are translated into proteins. This breakthrough was accomplished through a series of landmark experiments in the 1960s. It was in 1961 Marshall Nirenberg and Nirenberg-Matthaei conducted a groundbreaking experiment to decipher the genetic code. They developed a cell-free system by lysing E. coli bacteria, releasing the cytoplasmic contents necessary for protein synthesis. They obtained four different fractions, namely ribosomes, RNA from ribosomes, RNA from cytosol, and RNA-free cytosol as shown on the slide. Instead of fraction 2, that is RNA from ribosomes, they introduced synthetic RNA sequences such as polyuracil, poly-U, to determine their role in directing protein assembly. To trace which amino acids were incorporated into proteins, they prepared mixtures containing all 20 amino acids with one amino acid radioactively labeled using carbon-14 in each experiment. By varying the labeled amino acid across different trials, they could identify which amino acid was added to the protein in response to the synthetic RNA. Notably, when poly-U RNA was added, only phenylalanine was incorporated, indicating that the codon UUU specifies phenylalanine. This experiment was pivotal in unraveling the triplet nature of the genetic code and understanding how RNA sequences dictate protein synthesis. In analogous experiments with other synthetic RNAs, they found that poly-C directed synthesis of polyproline. In 1964, Nirenberg and Leder elucidated the triplet nature of the genetic code and allowed the remaining ambiguous codons in the genetic code to be deciphered. While Nirenberg and Matthaei first cracked the code for UUU phenylalanine using long synthetic RNAs with mixed bases presented a challenge. The randomized sequences such as those rich in cytosine and uracil resulted in mixtures of codons like CCU, CUC, UCC that produced proteins containing proline, leucine, and serine, but without revealing which codon corresponded to which amino acid. To solve this, Nirenberg's group turned to very short synthetic RNAs. They found that the trinucleotide UUU, which is the codon for phenylalanine, was able to cause specific association of phenylalanine charged tRNA with ribosomes. This association could be detected by passing the mixture through a nitrocellulose filter. The filter captures ribosomes but not free tRNA. However, if tRNA was associated with the ribosome, it would also be captured along with the radioactive phenylalanine attached to the tRNA. They similarly found that trinucleotides AAA or CCC caused ribosome association of lysine tRNA or proline tRNA respectively. This breakthrough set the stage for a systematic decoding strategy. If each of the 64 possible trinucleotide triplets could be synthesized and tested in the filter binding assay with 20 different amino-acyl tRNAs, their corresponding amino acids could be identified. However, synthesizing every sequence-specific trinucleotide was a monumental task. Leaders' pioneering studies used trinucleotides made by breaking down long random poly-GU RNA with the nuclease and purifying specific trinucleotides by paper chromatography. He determined that GUU, UGU and UUG encoded the amino acids valine, cysteine and leucine respectively. Further, Nirenberg's group, assisted by DNA and RNA polymerases, devised methods to produce specific trinucleotides such as AGU and UAG using polynucleotide phosphorylase and RNase A under methanol conditions. Finally, Merton, Burnford and colleagues employed these techniques to identify other codon amino acid relationships. UUU and UUC for phenylalanine, UCU and UCC for serine, and CCC and CCU for proline, highlighting the redundancy in the genetic code. Countless researchers in Nirenberg's laboratory and beyond then contributed to mapping the full genetic code. Nirenberg was awarded the 1968 Nobel Prize in Physiology or Medicine. He shared the award with Har Gobind Khorana of the University of Wisconsin and Robert Hawley of Salk Institute. Let us look into the salient features of genetic code, which are as follows. The genetic codes are triplet in nature. That is, 3 nitrogenous bases comprises one genetic code arranged in a specific sequence. They are commaless. There is no comma or any punctuation mark between the adjacent genetic codes. They are non-overlapping. On the mRNA, one codon do not overlap with the next codon. And they are universality. Same genetic code is present in all organisms, including virus, bacteria, and all living organisms. Degeneracy of genetic code. As the number of genetic code is more than the number of amino acid, it is implied that one amino acid may be coded by more than one codon. This is known as degeneracy. Ambiguity. A genetic code is always non-ambiguous. That is, one codon never code for 2 different amino acids. Collinearity. The codons on mRNA and the corresponding amino acid residues in the polypeptide chain have a linear arrangement. And here you can see the genetic code table. Thank you all.